We left off here as we were starting to talk about ribosome binding sites instead of promoters, or in addition to promoters, as an option for controlling gene expression. Ribosome binding sites are smaller, and they give us this ability to modulate protein translation directly and over 6 orders of magnitude. So there's some advantage in targeting them rather than a promoter. Of course, because an RBS is on RNA, we have to think about a number of things related to the sequence context beyond just the AG richness or the sort of proximity in sequence space to the Shine-Delgarno sequence and to the start codon. So what we're gonna talk about next is the idea that the RNA, which we've already discussed as having some secondary structure that's important, factors into these kind of calculations related to translation initiation. The simplest way to think about this is one extreme where you have, say, a strong RBS or an RBS in general, and then you also have a really strong region of secondary structure. So you've got the single-stranded RNA, and it has that consensus Shine-Delgarno sequence, but then it has another sequence that's complementary to that, and it just kind of folds up in a hairpin and maybe gets tucked in to a region that might look like it's buried in one of these stem loops or hairpins. In that case, the ribosome can't really see your RBS, right? So we know that the sequences around the RBS, and to some degree even somewhat distantly positioned, can play a role in just whether the ribosome can find the RBS. In addition to that, there's a number of things once translation is initiated that govern the speed and rate of translation. We focus on translation initiation because it's rate limiting. But in the process of translation elongation is very important in terms of getting proteins to fold a particular way. So there have been studies that have shown, for example, that the ribosome may ramp up or ramp down in particular regions. We know this empirically because in the RNA region, in addition to its secondary structure, are the codons that it encodes. And we know that even if we have a really strong RBS, if we use very rare codons in our gene sequence, then we can end up creating a situation where the ribosome stalls because there isn't enough of the tRNA that has the anticodon that would then bring an amino acid to continue reading that message. So one way that the RNA can impact translation elongation is just in the codon usage as it affects tRNA availability. Another, though, are other secondary structure effects, within the RBS, within the mRNA downstream of the RBS. So while a ribosome, once it finds an RBS, begins this process of translation initiation that really unfolds local structure, you can still have structure downstream that typically slows down the ribosome. I guess if it's really streamlined structure, it can also speed it up. And you can imagine that a certain amino acid sequence that's made as a peptide chain is elongating might, if made very quickly, fold into a state, a conformation that's more like a kinetic trap, rather than if made really slowly, finding the local energy minimum, its thermodynamic optimum. So these are ways in which mRNA can influence translation rate and protein expression. So in general, though, as we come back to the RBS calculator and focus just on the RBS for now, you want to provide any kind of calculator or manual approach that you might be taking to calculate RBS strength with some sequence context besides just the 7 or so nucleotides that are in the Shine-Delgarno sequence. And so here's a visualization. We've seen some before of RNA secondary structure. There are online calculators that can predict this, just again, from the RNA's perspective, trying to minimize energy. And you see these loops all have different kinds of names. And you can imagine that if the RBS were in some parts of this RNA, it would be problematic. So when it comes to RBS calculation, you've got this structural effect and you've got the strength of the RBS, which, because every organism has a different 16S rRNA consensus, or 16S rRNA sequence, I should say, leading to a different consensus Shine-Dalgarno sequence. Any sort of calculation approach is organism specific. But the 16S rRNA regions are pretty well documented. And so you can really program a biophysical model to be able to know what those interactions are, to be able to map interaction strengths of particular base pairs within RNA, and then to be able to do an RNA secondary structure calculation. And so that's a very oversimplified description of what the RBS calculator developed by Howard Zallis when he was a postdoc in Chris Voigt's lab is able to perform. I think whether or not we dive into any more details of the model, something that we can take away is that this model has been used a lot. This is a free online tool, and when characterized initially or in its second sort of iteration with some improvements, there have been experiments like we talked about last time, often just measuring fluorescence here on the y-axis over 6 orders or more of magnitude. And then you've got translation initiation rates that are predicted by the RBS, and you end up with this really strong R-squared value, where when you look at the translation initiation rate error cutoffs, 93% of the sequences that are predicted have less than 10-fold error. So that's pretty impressive. And so in your homework, you get to play around with the RBS calculator. Hopefully you'll find it a useful tool, I think, for a lot of metabolic engineering work. It's really valuable, particularly in bacteria, where when you have an operon, you can modulate individual RBSs.